In 1984, a Florida road crew unearthed human remains from a bog and called police. It looked like a homicide. It wasn't. Those remains were 8,000 years old, and 91 of them still contain preserved human brains. Deputy James Cox arrived at the bog just after sunrise, boots sinking into the muck where a road crew's backhoe had turned up a human femur. Police tape went up in minutes. The scene had all the markings of a possible crime: bones tangled in black peat, no sign of clothing, and a silence broken only by the hum of heavy machinery idling nearby. Dr. Robert Anderson, the county medical examiner, crouched at the edge of the pit, brushing away wet clumps of peat. He expected to find evidence of foul play, maybe a recent homicide. Instead, he found joints locked tight, bones stained deep brown, and no trace of fresh blood or tissue. The texture of the remains was wrong for a modern case, too rigid, too dark, too intact. Anderson called for the state archaeologist. Within hours, the site shifted from a police investigation to a controlled dig, with the medical examiner's initial skepticism giving way to careful sampling and forensic documentation. The question hung in the air: how could so many bodies end up here, untouched by time, and what secrets did the bog's chemistry hold beneath the surface? Radiocarbon dates from the first test samples stunned the team. Accelerator mass spectrometry placed the remains between 7,000 and 8,000 years before present, long before the rise of the pyramids, older than Stonehenge. The scale of the site came into focus as peat was peeled back in careful layers. Excavators documented 168 individuals, each buried with deliberate care. Some were wrapped in woven plant fiber shrouds, others positioned in a fetal curl, their bodies pressed close to the ancient pond's edge. The sheer number of burials, all clustered within a single acre, spoke to a community returning to this place again and again over centuries. These weren't scattered accidental deaths. This was a planned cemetery maintained across generations. The evidence of textiles, grave goods, and repeated use transformed the site from an archaeological curiosity into a window on the social lives of people who lived in Florida at the dawn of the Holocene. The question of how such delicate remains survived for so long would soon take center stage. The chemistry of the Windover Bog created a barrier against time itself. Beneath the surface, the peat's acidity hovered near pH 3.8, strong enough to denature enzymes and halt the usual decay of flesh. The acidity was a powerful preservative. In this oxygen-starved world, ordinary microbes that feast on tissue stood little chance. The microbes could not survive. The waterlogged peat, thick with humic and fulvic acids, formed a gel-like matrix that locked out air and immobilized cells. The gel-like matrix sealed everything. Iron dissolved in the bog bound tightly with these acids, creating phe-humic complexes that acted as natural preservatives, suppressing the chemical reactions that would have destroyed even the most delicate brain tissue. The iron created a protective bond. Dr. Michael Buckley, a specialist in ancient biomolecules, describes this as a chemical shield, a rare convergence of acidity, anoxia, and iron chemistry. The chemical shield was unique. In these conditions, the biological clock slowed almost to a stop. The biological clock nearly halted. Instead of dissolving into the earth, brains and other soft tissues were stabilized at the cellular level, their structures protected for thousands of years. They remained intact for thousands of years. This unique cocktail of acid, water, and metal set the stage for the extraordinary survival of prehistoric human brains, making Windover unlike any other burial site in North America. It was an extraordinary preservation. Preserving the ancient brains was only the beginning. In the lab, Dr. Ken Wachter faced a challenge that few had ever tackled: how to extract genetic material from tissue that had spent 8 millennia locked in acidic peat. Standard protocols failed against the bog's chemical arsenal, humic acids, iron, and degraded proteins. Wachter's team turned to scanning electron microscopy, revealing that the brain cells retained their basic structure, but the DNA inside was fragmented and tangled with inhibitors. They designed a new extraction pipeline, starting with a silica-based lysis buffer strong enough to break down the peat's complex matrix. Each step was performed in a sterile hood using single-use tools to prevent contamination. To isolate DNA fragments often shorter than 100 base pairs, they adopted a single-strand library protocol originally developed for ancient bone, but now applied to soft tissue. The method allowed them to recover endogenous DNA at rates above 30% in some samples, a remarkable figure for remains this old. This breakthrough opened the door to sequencing not just mitochondrial DNA, but also fragments of the nuclear genome, setting the stage for a new kind of ancestry analysis that would soon rewrite the story of Windover's people. Genetic results from the Windover brains reveal a story written in kinship and ancestry. Mitochondrial DNA from the site falls into 4 Native American haplogroups: X2A, B2, C1, and A2, lineages still found among southeastern tribes today. Analysis of the nuclear genome, even at low coverage, places these ancient individuals within the same genetic cluster as later archaic peoples and modern descendants along the eastern seaboard. Family links run deep across the peat. Kinship studies identified first-degree relationships with parents, children, and siblings buried just meters apart over a span of 13 centuries. This was not a random collection of remains, but a community returning to the same pond, generation after generation, to lay their relatives to rest. Textiles woven from plant fibers wrapped many of the dead, some adorned with strings of shell beads or tools placed beside them. Patterns in the burial pit show that children and elders were cared for. One grave held a child with a congenital disorder, buried with what may have been a comfort object. Among these signs of compassion and craftsmanship, evidence of darker moments also surfaced. The remains of a young man bore clean-cut marks at the neck, his head buried separately from his body. Forensic review found no signs of battle injuries on the others, suggesting this was an isolated act, personal, not warfare. Windover people left behind a record not just of survival, but of ritual, artistry, and the full range of human bonds and conflicts. Windover's preserved brains now yield ancient genomes, rewriting what we know of America's first peoples. As science deciphers their secrets, our oldest mysteries become tomorrow's revelations. What might we uncover next?